Hey guys, this is Mrs. Hallman for part 3 of the cell cycle and cancer disorders lecture for our cell division unit. This is the third and final part. We left off talking about HPV and how there were genetic or how there were cancerous strains of HPV, like HPV 16 and 18. The way that those viruses actually cause cervical cancer is that the virus will infect the epithelial basal cells of the cervix, and they will go through a normal viral replication cycle within a few weeks. And the HPV will actually insert part of its DNA or integrate part of its DNA into the cells of the cervix. That integration has a small chance of developing into cervical cancer, which is why there's a strong connection between viruses and cancers. And that's not the only viruses that is the only virus that's connected to cancer development as well. In this picture, we're now kind of investigating, we're moving away from cancer, which is the division of cells that we don't want to divide that continue to divide, and we're looking at the other side of this now. So what happens when we want more cells, but we can't always get more cells to divide, and how we can apply stem cells to help solve that problem. So you can see all of the different applications of stem cells to cure all of the diseases that are shown in this image here. So definitely a really useful tool that we've been able to apply. So what are stem cells and what do they do? Well, there's a bunch of different classifications of types of stem cells, and it all comes down to what types of cells can they regenerate. So up at the top, the granddaddy of all stem cells are called totipotent stem cells. And we biologists are not creative when we name things. Toti kind of sounds like total, and potent has to do with how well it's able to infiltrate or how well it's able to spread. So totipotent stem cells are able to create or differentiate into all of the types of cells, so total of all of the cells, totipotent. They can also regenerate themselves. Totipotent stem cells can also make pluripotent stem cells, which we'll revisit in development a little bit later in this presentation. Pluripotent stem cells can make more of themselves, or they can differentiate into different types of stem cells, which we typically call oligopotent or multipotent stem cells. And those stem cells can differentiate into one or maybe a couple different types of stem cells, but they're pretty far developed themselves. They can create these things called progenitor cells, which would develop different types of tissue, such as muscle, nerve, bone, or any other type of tissue in our body. So you can see here's the whole list. We have totipotent stem cells at the top, pluripotent that make and differentiate into a couple different types of stem cells, and we'll see what that looks like in a second. Multipotent that can differentiate into a number of cells, but usually very closely related in anatomy to the type of cell or the family of cell that it's in. Oligopotent, which can differentiate only into a few types of cells, and then unipotent, which can only produce one type of cell or themselves. So if we look about where we get these actual cells from, you can see that the whole process begins with fertilization with our egg and our sperm. And after 1 or 2 days after fertilization, our cells have merged and they've started to begin dividing. After that division has happened a couple times, we enter into the stage called the morula stage, which is a bundle of cells, kind of looks like a raspberry, of about 12 to 15 cells. All of these cells at this point in the morula stage are totipotent stem cells, which means they can develop into any type of cell. If we advance a little bit past that, we reach the blastocyst cell, which is a little ball, a little hollow spherical ball of cells. And in the center of that, we have this little bundle of cells in the center. We call them the inner mast cells. This outer bundle of cells will develop into the placenta, and this inner bundle of cells will develop into the human fetus. So these cells here, I have no idea why they're blue, but these cells are pluripotent stem cells. So they can in turn differentiate into all types of adult tissue with the exception of these cells that will develop into the placenta. So you can see how pluripotent stem cells would still be pretty useful because they can make anything except the placenta. Those pluripotent stem cells would then develop into multi and unipotent stem cells that are more differentiated, more specified towards what that cell needs to do. We divide our stem cells into either embryonic stem cells or adult stem cells. Adult stem cells can be harvested from areas where we have a lot of stem cells. So for example, in our bone marrow that produces red blood cells or erythrocytes, we can harvest blood cell or stem cells from there to help treat patients that have leukemia. Embryonic stem cells are harvested from that blastocyst stage, so it means that that embryo is terminated when we harvest those cells. Those cells can then be plated and they can differentiate depending on what condition they're in into a multitude of different types of cells, like liver cells or potentially nerve cells. There are some ethical issues with embryonic stem cells simply because the embryos, once you harvest the pluripotent stem cells, will never develop into an embryo, which makes adult stem cells a whole lot more attractive. However, they present a bunch of different challenges with research. For example, adult stem cells are found in a lot lesser quantity, so it's a lot harder for us to obtain them and then to filter out all of the normal cells and to purify just the stem cells. They're also a little bit more restricted in that they can't develop into as many things, so it's a lot harder to work with them in an experimental condition. They also have been exposed to a lot more environmental hazards, so they might have been exposed to more sunlight or UV radiation, more toxins, more carcinogens, which might make the DNA a little bit more vulnerable towards mutations, which is not an ideal treatment or not an ideal cell to use for treatment. So with this, there's kind of the back and forth between the use of adult stem cells and embryonic stem cells. You can see kind of summarized here, we've been using adult stem cells for a while now. We have tons of therapies that we apply adult stem cells, but it always comes down to the fact that they are not nearly as applicable as embryonic stem cells simply because they're multipotent instead of pluripotent. The embryonic stem cells or early stem cells, there are some issues with that. There was some legislation that said that embryonic stem cell research could not receive any federal funding. And since this slide was created, we've actually had another executive order by President Obama in 2009 that has released that restriction. So stem cell research on early stem cells is now permitted. They do receive funding, but there are two foundational rules that still apply. And those two rules are that we cannot create excessive amounts of embryo for the sake of creating them for experimentation. So in other words, we can't create embryo farms where we're just pumping out a bunch of these for the sake of experimentation. And secondly, that we can't be cruel or inhumane with how we treat these embryos. So there has to be a little bit of respect. We cannot torture or mutilate or harm these embryos in our experimentation. These stem cells, we've used them to help fight arthritis and inflammation involved with the interaction of arthritis. We've used them to fight apoptosis and cell death in spinal cord injuries, and we've also used them to develop different types of tissue like bone and muscle and cartilage. The embryonic stem cells that we harvest from the blastocysts, those are also used in addition to the umbilical cord stem cells that you can harvest from the umbilical cord of a developing baby. Adult stem cells would be harvested either from bone marrow, and there's a really good video on the next slide that shows that process happening, or they can develop anywhere else that we have adult stem cells in some sort of quantity that we can extract and purify. Those adult stem cells that are coming specifically from bone marrow are donated from people who want to help other people who have either lymphoma or leukemia, and those bone marrow stem cells are extracted and then purified and replaced into the patient. So there's a great video on that here about how chemotherapy radiation is applied after stem cells have been harvested and then used to treat lymphoma or leukemia. And last are just two links for you guys. The first one is the National Institutes of Health for Stem Cells, so a great place to start if you're looking for stem cell articles. The second link is the Cancer Genome Atlas that basically goes through and categorizes and classifies and kind of informs you about different types of cancers. So that is the end of the cell cycle disorder lecture, and I hope that was helpful.